hello viewers. Welcome to Research Digest podcast. We are starting a brand new season of this podcast, and in the first episode, we have Dr. Deena Al Mahbuba with us. Dr. Mahbuba is currently working as a senior scientist in AbbVie Incorporation. Apu, welcome here. Thank you so much for coming to my show.
Thanks for having me.
Okay, first, let's start with your career journey. So, can you please share briefly where did you start and how did you come to your current position?
Sure. So I'm originally from Bangladesh, and I did my undergrad at University of Dhaka in biochemistry and molecular biology. And I followed up with the master's in the same subject in the same university. And from there, I went to UCL, University College London, to do my second master's actually in molecular medicine. And from there, I moved to US to do my PhD. I did my PhD at MIT at the chemical biology program in the chemistry department. My supervisor was Professor Laura Kisling, and I finished my PhD studying carbohydrates biologies and how it dominates how it directs the sulfate decision of the human pluripotent stem cells. And I finished my PhD in 2022, summer of 2022. And from there, I stayed back in the same lab for a few months as a postdoc just to finish up my papers. And then I started my next stage of career as a senior scientist in neuroscience discovery at AbbVie. And that is at 2023, February. And I'm working there since then.
Okay, wonderful. So there was a kind of little bit switch of your field, right? From
Yes.
carbohydrate biology from neuroscience.
Yes, there was. Actually, there was a little bit more because so my undergrad was in biochemistry and molecular biology. So that was a pretty big umbrella that covers a lot. And when I started my master's in Bangladesh first, and I decided to do it in immunology, I was like, okay, like everyone else, I'll do my master's research in immunology, and that's where I started at ICDDRB with Professor Kadri's lab. And I followed the same field when I went to UCL to do my second master's. That was also in molecular biology, but focused in infection and immunity. But at some point of my career, I decided that that's probably not what I wanted to do for my rest of my life. So I decided, like, okay, I think I'm more interested about more basic biology to understand more about the development and development and disease. And stem cell was quite a bit of a hotspot at the time, and I was really fascinated about stem cell biology, and I happened to had a chance to work in a lab that does work in stem cell biology. So that's
Right.
where my interest and opportunity
A
came
lot of
together.
different fields. Yeah, you covered a lot of different fields. Yeah,
Yeah.
things changes, but I think what matters is your love for science and your intention to do more science.
That is
Okay, so
true. I think like down there, like in the road, I realized like I really like basic science, and that was my biggest motivation to move to more into
so it's more
developmental
fulfilling,
biology.
right?
It was, yeah, it was.
Okay, thank you so much for sharing your journey, Apo. And the main article that we are going to discuss today was published in ACS Nano in 2023, and the title is Dynamic Changes in Heparan Sulfate Nanostructure in Human Pluripotent Stem Cell Differentiation. So, uh, you already guessed, we'll talk with the first author, Deena Al Mahbuba, regarding mainly this article. But more recently, this same group has published another article, which is in Stem Cell Reports. So we'll get some information from back and forth, hopefully. Okay, so let's start up. So how did you really start this project? So tell us briefly, how did you start, and then we'll go into other details.
Sure. So the lab I started my PhD in, the focus of the lab was to understand the carbohydrate or sugar, basically. And one biggest reason for that is sugar is everywhere, but there is very few in terms of technology, in terms of science. The information about sugar biology and its role in development and all other different aspects of life is quite limited. And that's the motivation where my lab comes into the like, okay, we will study sugar. My professor, Professor Laura Kissing, she is a pretty big name in the sugar biology. She is herself as a chemist, and I happened to do my PhD in her lab. So that was my intention to that gave me the that gave me the opportunity to work in sugar biology. I was particularly interested in stem cell biology, and as a sugar biology lab, we used to study in different impacts of sugar in different scenario, like in stem cell microenvironment, in infection and immunity, in the bacterial biology, in all sort of things. So I particularly picked like, okay, I'll study sugar in the stem cell biology to combine my interest and the lab interest together.
Okay.
So that's sort of where it began. And I was, even before me, there are a few other folks from the same lab who study sugar biology in the stem cell microenvironment. And there are a few really nice papers published by the lab that sort of like set up the ground from where my work stemmed out.
Okay, got it. So most people work on cell surface proteins, and I think very few people work on cell surface polysaccharides. So that's really interesting. So tell us more about the role and the importance of cell surface polysaccharides and how they play a role in the bigger aspect of the biology.
Sure. You are absolutely right. The community is very small, and as I was saying, like it was quite a bit of a niche. Um, for a long time, I think and I would say like people who are not in the field, they still think carbohydrate as like an energy source. Like when I say someone like, oh, I study carbohydrate in my PhD, they're like, oh, did you study it as a source of energy or as what, how exactly you studied it? So we try to look at carbohydrate more as a biomolecule and understand how this carbohydrate can contribute to different aspects of cell fate decision, understanding the cell, what's its role in the cell to cell signaling processes. The reason because of that is if I go take a step back and talk about a little bit where this carbohydrate is coming from. So extracellular matrix for a long time, it has been studied as like, oh, it is an inert scaffold system that just gives us like passive support system for the cell. But throughout a long research, we identified not necessarily. They have so many biochemical and physical properties that can modulate how the cell behave. For example, in case of the stem cell perspectives, it can modulate how the stem cell differentiate or how the stem cell basically function. A big portion of the ESM is actually coming from the glycocalyx, which is the sugar coat of the cell. And it's everywhere. All of our cell in our body is covered with a sugar coat. And as I was saying, like it was for a long time was studied like, oh, it's an inert, passive, mechanical thing that covers the cell and gives the support to the cell. But we identified not through our lab only, but throughout the research for the last couple of decades, it has been identified that like no, sugar has the ability to actually modulate different signaling processes. And particularly the sugar that I was interested very much was the heparan sulfate proteoglycan, which was perhaps the most influential GAGs. It is a glycosaminoglycan, one of the mostly adequate sugar on the cell surface, part of the glycocalyx. And like, even if I talk about the very recent findings, is like one of the findings was that like, oh, heparan sulfate as a glycocalyx can modulate how the pluripotency of the stem cell is maintained. Because even from our lab, we showed previously that the pluripotent cell can bind to the heparin binding portion of the heparin binding peptide, to the heparin of the presented heparin, and that can influence how they will propagate, or that can influence how they will maintain their pluripotency, and that also can influence, eventually we showed that how this pluripotent stem cell actually decide their fate decisions. So that's like where the interest was there. We wanted to study sugar, most like, can we study sugar as a biomolecule that influences different signaling processes? And what are these signaling processes? How does it do exactly?
Right. Okay. So the specific glycan you chose for your study mainly focused on heparan sulfate, right? So tell us more about heparan sulfate. So, and why it's important to study their distribution, and let's say like more specifically spatial distribution, and what might be their importance. Yeah.
Sure. So heparan sulfate proteoglycan, or HSPG. So it is one type of GAG, so glycosaminoglycan. So it is probably one of the most prevalent GAGs on the cell surface glycocalyx. And one interesting factor about the heparan sulfate, just being as a carbohydrate, it is very complex in nature. It is composed of a simple disaccharide unit. So it's a uronic acid and the glucosamine, like alternative disaccharide unit. But it can be very long. It can go from 20 to 150 nanometer long, and can made of like up to 400 sugar residue. It's always depend from a protein core. That's why it is called a proteoglycan. And one interesting factor is that even if it is a simple disaccharide unit, it can have a very complex and heterogenic structure, because it has the ability to go through a lot of post biosynthetic modification, like sulfation, epimerization, and N-acetylation, a lot of this thing. And as we know, that carbohydrate is sort of like a secondary gene product, right? It's not like protein or nucleic acid that is built on there is a genetic code that directs how the proteins are coded and everything. Sugar biosynthesis is much more complex. It depends on so many other factors, even like the metabolic state of the cell can direct what sugar modification you're going to have. So in general, just by being a sugar, it can have a very, very complex and heterogenic structure. And it has been shown from a lot of previous literatures in the field that heparan sulfate can go through a lot of diverse range of changes. One prominent factor was it can go through a lot of different sulfation pattern. Like there's certain types of cell, like in A state, they can have a certain sulfation pattern. When they move from A state to B state, they can have a different sulfation pattern. But always the question was there, why? It's a lot of change to do for a cell, but why? And eventually it has been shown that, heparan sulfate can act as a coactivator or as a signaling factor or activator of a lot of different signaling pathways, like very prominent important signaling pathways like FGF, BMP, Wnt signaling pathway. All of these signaling pathways, they're very critical for the stem cell pluripotency and also stem cell differentiation. So there was so there are two factors that was very critical, that heparan sulfate proteoglycan can have a very diverse differences in its nanostructure. That we know now, but at that time it was heparan sulfate structure can go through a lot of diverse changes. And we know the other piece was heparan sulfate can act as a activator of a lot of different signaling pathway. Right. So understanding was does this structural changes, how the structural changes are impacting this functional perspectives? That piece was unknown. We didn't know. We didn't know much about it. There was a big knowledge gap. So that's sort of where we wanted to understand, and in the aspect of the human pluripotent stem cells specifically.
Okay, wonderful. So to study this particular thing, you used expansion microscopy, right? So
Yeah,
you did some modification to that protocol. Though, I know it's a technical question, but
sure.
can you share some insight on that? What kind of modification you did to that expansion microscopy? And yeah, sure.
So expansion microscopy, if I try to tell you in a very brief way, so the concept of expansion microscopy is like, well, in terms of if we go to just the microscopy, the idea of microscopy is like you are using the light and energy
Mhm.
and optical machineries to visualize the cell, right? And you can use different magnification to see, like basically blow up the cell and see more detailed structure and everything. So the concept of expansion microscopy is, instead of blowing up your magnification of your physical instrument, you are actually try to zoom in the sample itself, or I should say, like blow up the sample itself in a certain way, so you can zoom in with your regular microscopy, but you can see more detailed structures and information there. That's like
Mhm.
the very basic concept of expansion microscopy. Dr. Ed Boyden, he's the pioneer of expansion microscopy, who works at MIT. He used to work at MIT Media Lab. Now he works at Brain and Cognitive Center at MIT. So I work very closely with him on this project. So for when expansion microscopy, I believe it came in light around 2012, 14, around that time. And it was heavily applied to understand the protein biology, protein-protein interaction, and how the co-localization of protein
Mhm.
more in the concept of the brain biology. And then it was applied to the nucleic acid biology to look at the nucleic acid localization and so on and so forth. So before we worked on this, there was not much. Wasn't much data available. Like, can we use expansion microscopy for the glycan to look at the carbohydrate or sugar? There is one point. There is a one very specific reason to it, and I think it's still that knowledge gap is still not completely covered. For that, I have to explain a little bit of expansion microscopy. So what happened in expansion microscopy is basically you're trying to blow up your sample, right? So the idea is like you kind of embed your sample in a polyacrylamide gel, and with specific anchors to attach to different proteins. And when the polyacrylamide gels are formed into the protein, into your tissue or into your cell culture, they sort of, that acrylamide gel sort of like attached to those anchors, and that is how it sort of like that is how the whole tissue or cell culture gets embedded into the polyacrylamide gel. And then you use some proteinase K to chop it off, because when you're gonna try to blow it up, which you do by
Mhm.
adding the water to it, then it can actually move. Especially it can move it can move from each other, and it can just gets easier to zoom into it. So one key factor is you have to use the anchor that binds to the protein, right?
Okay.
And that's a very specific chemistry. And
Mhm.
that is critical when you're trying to look for a specific biomolecule. For carbon, if you want to have just a carbohydrate chain that you can want to visualize, so you need to find an anchor that actually bind to that carbohydrate. But that's chemistry is still sort of missing. Like, we do not necessarily have access to that chemistry just yet. There is
Okay.
a lot of work going on, and I should keep it on, because like my some fellow colleagues in the same lab that were there actually trying to figure this out, and it's still not there yet. So
Okay.
what we did here, because heparan sulfate is a proteoglycan, so it does have a protein core, so we
Mhm.
use that information to do expansion microscopy with the glycan. So what we did is like it's a pro EXM, so it's a protein retention expansion microscopy. So where we use the same anchor molecule that can attach to the protein core of the heparan sulfate proteoglycan, and that is how it is attached to the polyacrylamide gel. The reason this is very critical because you have to kind of somehow preserve the spatial distribution, because when you blow it up, you have to keep it sort of intact. It's more like you draw something on the balloon,
Mhm.
and when you blow up, it expands, but it keeps the image intact. So it's something like that. The anchor works like that. Like it's keep it intact. It just gets blowed up and separated to each other. So
Oh,
it's like zoomed in version of it. And that's why that's why the anchor is very critical. So for heparan sulfate imaging, we use that strategy to use, like, okay, we will just use the same anchor that will bind to the protein core of the heparan sulfate, so it stays the spatial distribution intact when we blow up the sample. But some follow-up work is going on where they're trying to understand and develop some chemistry where they can bind directly to the carbohydrate, so they can basically use this expansion microscopy to visualize more carbohydrate molecules. And there are some other future thought there.
okay.
So yeah.
Okay, wonderful. So you use this technique to see the heparan sulfate, and also I think you observe some pluripotent stem cells, and particularly got some really critical findings when they kind of uh, became a motor neuron cell, right? So from the pluripotent stem cells. So what were your major findings and kind of key observations?
Yeah. So our first interest is like, oh, okay, can we use expansion microscopy to visualize the carbohydrate? And
Mhm.
if we do, what does it tell us? Because even with the conventional microscopy, we can see the expansion. We can see the heparan sulfate staining around the around the cell colony, basically. So I don't have the image here, but if I can show you, like when you have a pluripotent stem cell colony, and when we use the conventional microscopy to visualize it, we usually see the heparan sulfate staining just around the edge of the colony. We most
Mhm.
of the time do not necessarily see it in the middle of the colony. So we were like, okay, but it should be present for all of the cells, but we can't necessarily see it, probably because the nature of the pluripotent stem cell colony is like it's very compact, it's tight and packed. So our first thought was like, okay, can we visualize individual cells, and can you
Mhm.
visualize the heparan sulfate around it? And we first saw that, and it was a very striking data because we see such a detailed structure of the heparan sulfate. And the key findings was from that specific experiment was like, okay, we see heparan sulfate has a long protrusion hair-like structure, and it's all over, it's covering pretty neatly and nicely all over the cell. That's actually sort of very nicely presented in a way that it can interact with a lot of other macromolecules from the extracellular milieu, and a lot of it has the possibility also to interact a lot of other growth factors and everything. So it's like very extended. So because the nature of the pluripotent stem cell is has the ability to differentiate to all different types of cell in our body. So it can differentiate into the ectoderm lineage, which eventually give rise to neurons, glial cells, our pigment cells, neural crest cells, and so forth. It also can give rise to mesendoderm and endoderm lineage that gives your kidney, lung, like other sort of cells.
Mhm.
So we want to say, okay, this is how the heparan sulfate looks like in pluripotent stem cells. And we do know that heparan sulfate can, at least from the literature, we know, like from the analytic analysis or biochemical analysis, we know that the sulfation pattern can change in the heparan sulfate when you differentiate into different lineages. So we were like, okay, does it also translate to its nanostructure, like this long extended structure that we are seeing on the surface of pluripotent stem cell? Does it also go through any sort of changes? So first we differentiated the cell to the mesendoderm lineage, and which is also multipotent in nature because it can give rise to quite a different types of cells. And we saw, okay, it looks pretty much the similar. Biochemical analysis also confirms that they look quite similar in terms of length and pattern.
Okay.
And then we were like, okay, if we go to a different lineage, which is our last lineage, the ectodermal lineage. So we differentiated the cell to ectodermal lineage. We differentiated them first into the neural crest. And the neural crest is also a little bit of multipotent in nature. It is not a terminally differentiated cell. And interestingly, we saw like, okay, the cell, heparan sulfate looks different. They looks more like a dot, more like a puncture structure. Then we differentiated them even further. Like, okay, we will what happened if I differentiate the cell to the terminal end? Like, so I picked the spinal motor neuron differentiation. I picked a chemically defined protocol to do that. So, okay, we will differentiate them all the way to the terminally spinal motor neuron and see what the heparan sulfate looks like. And we saw it's strikingly different if you put the pluripotent and the spinal motor heparan sulfate side by side. The spinal motor heparan sulfates are very puncta-like structure. It's across the axonal body, and it's all over the cell. But even if you try to visualize it, just from seeing it, you can tell like there is a lot less number of heparan sulfate on the human motor neuron compared to the pluripotent stem cell state.
Okay.
So that was a big finding for us. Using the strategy that we developed in our lab or modified in our lab, I should say. And so that was a very interesting findings. But the obvious question that follows up, like, why? Like, what does it mean?
Exactly.
Okay, does go through some changes, but the basic question, like, why it does that? Because that's quite a lot of a change. It must have something to do with something to do that.
Yeah,
so we already
there's a reason.
yeah, exactly. And that's like, and we're there to understand that why. So that was very like a curiosity question, like, okay, why it does so. So we already knew that, okay, heparan sulfate can act as a co-activator of a lot of different signaling pathway. And one of the signaling pathway where it's already established that you can in the FGF pathway, which is very critical for the pluripotency maintenance and sulfate decision. And we know that heparan sulfate is a co-activator of that pathway. So that needs to be present when the FGF ligand binds to the FGF receptor. So we're like, okay, we will try to understand the function of that and function of heparan sulfate in that aspect and see if it correlates with these changes in the nanostructure. So we did I did a very simple experiment. I developed an assay. It's a FGF bound growth factor binding assay. And I wanted to see, okay, how the growth factor binding changes when the cell differentiate. So I saw that, okay, at the pluripotent cell, they have a very high binding to the FGF, the pluripotent stem cell. When they differentiate to mesenterom, the growth factor binding is pretty similar. But the moment you differentiate all the way to the neuron, it's significantly shifted. It's a massive shift happens and very less binding. But that was very interesting because at least from the genetic data, we also found that in the pluripotent stem cells, the heparan sulfate has a higher chances to be sulfated. And we know that the higher the sulfation is, the higher the I reversed it. At the pluripotent stem cells, and they have a less chance of have sulfation, less chance of being sulfated. But we know that sulfation sort of correlates how it binds to the growth factor. So when we look at the sulfation pattern and the human motor neurons, we see that the motor neurons have a higher chance of being sulfated, but they bind much, much less to the growth factor. So it sort of contradicts what we are seeing. So obviously the genetic information, it is based on looking at the sulfation biosynthetic pathways, basically. So those genetic data is not correlating to what we are seeing. So what we hypothesize is that, so we think that the change in the nanostructure is another way, like the physical nanostructure of heparan sulfate is another way by which it actually modulates its ability to interact with the growth factor. So when they're long and extended version, they're very optimally presented to bind to the growth factor. So they bind a lot of growth factors, even though they are less sulfated. But when it rolls down back to a punctal-like structure, so it limits its ability to bind, even though they're very sulfated in the human motor neuron, but its change of nanostructure limits its ability to bind to different specific growth factor. And this is quite interesting because the FGF signaling is very important to maintain the pluripotency of the pluripotent stem cells, whereas for the case of human pluripotent stem cells, it needs to decline, it needs to decrease. The FGF signaling needs to decrease for the differentiation towards the motor neuron. So we think that was a missing piece that we never thought about, that the physical nanostructure, like its physical spatial distribution and also temporal distribution, because it comes in different stages of differentiation, right? So we think that one piece was unknown before, which we tried to shed some light on with this work. Sorry, I took really long to explain the major findings.
That's wonderful. That's wonderful. It will be helpful for everyone to understand. And in your more recent work, the one I showed at the beginning, so you generated a cell line which has heparan sulfate deficient cell line, and you observed that pluripotent cells cannot differentiate into ectoderm or mesoderm lineages. So it was also a really interesting finding. Tell us more about that briefly.
Sure. So it was a collaborative work with with a colleague in my lab, and we published this paper together in Stem Cell Reports, and that was more into looking into the biology of the heparan sulfate in the stem cell in the fate decision of human pluripotent stem cell. I want to highlight one thing, that actually knocking out of heparan sulfate is. It probably sounds like, oh, you just knock out something from the cell. What's a big deal? Because everybody does it every day at this moment. But it was a little bit of a serendipitous discovery, because without any heparan sulfate, it's very little. Like, cell cannot survive at all. So when we the colleague who actually tried to make that cell line, she was heavily discouraged, like, oh, it's never going to be possible. A lot of lab tried, it just didn't happen, because human pluripotent stem cell, they just don't survive when you successfully knock out heparan sulfate. What was serendipitous here was that, like, heparan sulfate helps the FGF signaling, and FGF signaling is required for the pluripotent stem cell, right? I just talked about it. And you need a lot of heparan sulfate, a lot of FGF in your media to keep your stem cell alive. And actually, the commercial media that you get, they have a ton of FGF, which actually helped this heparan sulfate deficient cell to survive. Because even without the heparan sulfate, because they're like flashed with so much FGF, they somehow managed to ignore that, okay, we don't have the coactivator, we still will somehow survive. So that was a bit of a serendipitous discovery. But anyhow, so once we have the cell line, this is actually the only cell line in the whole world, in the human pluripotent stage, that doesn't have the heparan sulfate. And we actually collaborated with a lot of different labs who were very interested to understand, because the signaling path works a little differently between mouse and human, obviously. And there's quite a bit of work has been done, but mostly focused on the mouse embryonic stem cell, because the FGF signaling was a little different. But there was no other human pluripotent stem cell that doesn't have heparan sulfate. But anyhow, so once we have the cell line, like, the next interest was to understand, like, okay, can we differentiate them into different lineages and see what happens? So my interest was very much on the neuronal side. So I first differentiated the cell to the neuronal lineage. So for the neuronal lineage, I differentiated them to the neural progenitor cells or the neural crest cells. And I say, like, okay, they can differentiate to the neural progenitor cell. maybe a little bit less than the wild type cell, but they still can differentiate. They express all those markers and everything. But the interesting finding was more in the terminal side, because when you differentiate as like a terminally differentiated mature neuron, I see that there was a very, very dramatic change in terms of axonal growth. And what was very interesting is to see that, you need those, for neuron, you need those very extended axons and dendritic growth that actually interacts with each other, make the synaptic connection, and make the functional synapses, right? And when we differentiated this heparan sulfate deficient cell all the way to the mature neuron, we see they don't necessarily have those extended exons. They have the cell body. They express those markers for the cell body, like the beta tubulin and stuff. But if you look at their axonal growth, they're very, very stunted, short, and they do not have the ability to make any functional synaptic communication with the other component. So
That's a
that
huge
put us.
difference. Yeah.
That was quite a bit of a huge difference. Like, oh, so heparan sulfate does have something very important to do for functional synapse interaction. And eventually, we also actually, some background work was there, and we found that, okay, there are some particular proteins. And in fact, around the same time, some other groups published some data where they identified that heparan sulfate has specific role to play by some interacting with specific synaptic protein like neurologine and neurogene, and neurexine and neurologine, and it helps those interaction to make a functional communication between the two neuronal cell. So that was my key finding from that experiment. And in parallel, we also identified, like, okay, when it goes to the other direction, like the mesendoderm direction, what happens there? And we identified that, okay, it cannot go like, it's not like the ectodermal lineage, rather it's like stopped at the very beginning of the mesoderm differentiation, and it cannot go past like a very, very beginning point. And eventually we identified there are some specific growth factor signaling pathway, the nodal activin signaling pathway, where the heparan sulfate is playing a critical role that is impacting how it's modulating the mesoderm sulfate decision. So that's like very short summary of what we identified in that paper.
Okay, that's great. So let me ask you another thing, so which is your challenges. So is there any particular challenge you faced during your these experiments, this whole study, that you overcame? The reason I'm asking is because we usually publish the positive data in journals, and journals publish all positive results, but there are a lot of things going on that we don't report, and journals don't publish kind of negative data or many challenges. So is there anything, something particularly popping in your mind?
Yes. There was very stressful nights around in the lab. But one, like, few things that immediately actually come to my mind. One of them is about the stem cell culture, actually. So, like when I'm talking about the stem cell, you need to culture them. Like, you can't culture them directly on the plastic plates. You need a surface. You need to cover your plate in some way. And actually, what you are using as a surface it's very tricky for the stem cell, because the stem cell in general is a very sophisticated cell. They have the ability to just differentiate spontaneously, and just you lose your pluripotent stem cell and you have to start from scratch and all that. So the surface is very critical. So usually the surfaces that people use for stem cell cell culture, they like, few of them are like Metrogel is a very common one. Geltrix is a very common one. These are the surfaces people use. Like, even before that, people use like the mouse embryonic fibroblast like as a surface. For neuronal cells, a lot of time people use astrocyte as a supporting cells, and so on and so forth. But The metrogel, which is composed of like 18,000 different proteins, and it's basically like extracted from the mouse extracellular matrix. So it has like a wide variety of the complex protein,
Mhm.
which allows the surface to become like really cushiony and very comfortable for the stem cell to grow. But one problem with that was that metrogel has heparan sulfate in it. And
Okay.
for all the work that we were doing, like for example, the knockout line that we developed, we cannot grow them on the metrogel, because if we grow
Mhm.
them on the metrogel, we're sort of contaminating the microenvironment, right? Because stem cells are
Exactly.
very,
Mhm.
very they are very sensitive to their microenvironment cues, like what you're presenting them, that will direct how they're going to differentiate. And if you present them heparan sulfate, it doesn't matter. Like if you remove the heparan sulfate from the system, they will act on like, okay, we have some extracellular heparan sulfate and we will just carry on. So
Exactly.
that will basically contaminate your model system.
Mhm.
So there was like a simpler version of like purified protein from that material. One of them is like a vitronectin that was developed by and commercially available that you can use as a surface. But the problem with that, it's you can imagine like it's a single protein surface compared to the whole 18,000 metrogel,
Mhm.
80,000 protein compost metrogel. And for the neuronal differentiation, I had to differentiate the cell on the same surface for like 21 days to up to 28 days, which is a long time. And it's very
Yeah.
difficult to differentiate them just on a simple vitronectin surface. A lot of time it would happen that they would just like by 14 days or close to 20 days, they would just lift off like the attachment. They will just like all fly out and they just die.
Oh, okay.
So
It
it
was
was
very
very
challenging.
that was very tricky, and I was sort of stuck with that because with the veterinarian I
Mhm.
can add some other single protein like laminin, PLO, and that sort of things. But even after that, it was very challenging to continue the differentiation on a much simpler surface. And yeah, like in some time that also stressed the cell, and they would differentiate. You can tell like they're differentiating to the neuron, but they're a very stressed neuron. They're
Right.
not probably the most best representative
Healthy,
neuron that you
happy one.
yeah, not the happy one. And it was also tricky with the expansion microscopy because you want the culture very intact when you actually form the gel,
Mhm.
like when you actually embed the sample in the gel. And if your cell get lifted off during, because you sort of like add material, wash it off, add few more times, and a lot of time it happened, they just lift off, and middle of my you know last experiment, they just decided not to cooperate.
So
So
sad.
that was
Okay.
that was tricky.
Okay. Thank you so much, Apu, for sharing this.
Yeah.
And also, finally, do you have any particular message for my audience on career science? Anything?
Oh, yeah, sure. um, well, one thing I think I'm comfortable to tell the younger generation of my audience is that um, I'm circling back to what we started with, that I made a lot of changes in my scientific career,
Yeah.
and I never really regretted what I did at that moment. And I started with immunology, moved to molecular medicine, and then I did a little bit of structural biology, like a really hardcore biophysical chemistry, and decided, no, this is not my thing. I need to move. And then I found my niche, and I really landed on the stem cell biology, and that is something I continue to do right now. What I wanted to tell people who are like in their 20s, like, don't afraid to chase your passion. I think
Mm,
in science, this is something you will really need it. You need to explore a few different things to find what we really, really like to do, because I think science can be very monotonous, and can be very boring if you don't really like it. So you need to love it. If you want to do it really well, you need to love it. So don't worry too much to explore new things, and try to find what you really like. I think that's one thing I can say very comfortably.
That's a great suggestion, Apo. And thank you so much for coming here and sharing your work so nicely and with so much um, easy explanation, because science is hard. Research was very uh, difficult, deep, and you shared very nicely. Thank you so much. And also, the viewers, thank you for watching. And I'll see you in the next episode, hopefully. Bye.
And thank you so much, Banna, for having me. It was thanks for giving me the platform to talk about my science, and I really appreciate your time and effort. Thank you.
My pleasure. Thank you all.